So they have centrosomes. Centrosomes are little barrels, they're little double barrels of microtubules. All right? And I have no idea what the M is for there. Somebody got crazy with the printing. But when they give the instructors figures to put in our slides and stuff, I think they don't go through the final edit that the textbook does, because I always find weird little typo things on if I'm looking at them. Anyway, so the centrosomes, microtubules, they sit around and they wait until the cell's ready to divide. And then what the centrosomes do, there's actually they'll duplicate, so you get the microtubules assembled into two double barrels, and they go to opposite sides, and they shoot out microtubules, and they grab your chromosomes and pull them apart. That's how mitosis and meiosis work. Let's see. Yeah. So the centrosome is this whole structure. A centriole is each barrel is a centriole. All right. All right, the membrane-bound ones. All right, so I'll be really quick on these guys. I'm sure you've heard the story before. But if you do have questions, ask, okay? You might be fuzzy on it, or maybe you didn't hear the story before. All right, just let me know. The nucleus, that's where you find your chromosomes. What does the word chromosome mean? Chromo meaning colored. Some from Latin soma. Body, colored bodies. All right, that's what they are. All right, usually you can't see the chromosomes. You just sort of see dark, grainy stuff all throughout. You see that? That's called the chromatin, which literally means colored stuff. So scientists first saw colored stuff because it's stained. They're using stains to visualize the cells, but they didn't know what the stains stick to like we do now. What the molecules actually stick to now. And so they saw this colored stuff, which was the proteins and DNA in the nucleus. But when the cell's ready to divide, they condense into defined little bodies, and that's what we call them chromosomes. All right. This is what happens when you're a cell biologist teaching you stuff. The ER. Yes, isn't it beautiful? Endoplasmic reticulum. All right. It's called endoplasmic because where is it? In the cytoplasm, yeah. And reticulum, you know what that means?
Net.
Yeah, yeah, reticulum, it can be like a net. Basically, it means that it winds around, right? So they kind of wind around through the cytoplasm, all right? And if they have ribosomes, it's rough. And you see how the rough ER is kind of wide and flat, and it's got the spaces inside, all right? The smooth ER, it's a little more narrow, it's a little more tubular, right? Right? And it's also hollow, there's space inside. And you guys probably know this, because I know a lot of you have had anatomy and physiology and things like that. But whenever you have a sort of an open space, even the Golgi, see the open spaces inside these structures? Whenever you have an open space inside of a biological structure, what do we call that? Well, they're called cisternae, but the space inside has its own name. The lumen, just like your intestines, right? The intestinal lumen, right? Your lungs have a lumen, right? Pulmonary lumen. Yeah, so it's the space inside. That'll become important later on. Then there's the Golgi complex. Well, the rough ER, it makes proteins, but so see how there's ribosomes kind of everywhere? So there's free ribosomes, they're making proteins that will stay in the cytoplasm. If they make them at the rough ER, they go inside the lumen of the ER, and then they pinch off and they get transported somewhere else. Right? And in little vesicles, they're called, little membrane-bound bubbles, vesicles. And vesicles have a lumen too. There could be proteins in there. And the smooth ER doesn't make proteins, it makes lipids. Yeah. So, and both of them send little vesicles to the Golgi, and the Golgi has enzymes in it that will modify the lipids and the proteins. They'll add carbohydrates to them, they'll add phosphate groups, they can clip off pieces of a protein, shorten it, things like that. Yeah, but so basically it does chemical modifications and these chemical modifications essentially become destination tags. It's like you're supposed to go to the mitochondria, you're supposed to be a lysosome, right? You're supposed to go to the nucleus, okay, things like that. You're supposed to go to the membrane. So we call it the post office of the cell, right? Because it packages and delivers things. Lysosomes contain digestive enzymes. If something fuses with the lysosome, they get degraded. All right, we'll look at that when we see active phagocytosis. Vacuoles can store food in cells, provide support, that turgor pressure I mentioned earlier. Mitochondria. I always thought mitochondria was a pretty beautiful word too. This is my daughter, mitochondria. All right. But mitochondria, I remember if I told you guys this. You know what mito means? No, that's a good one. I haven't thought about that. But no, it's not mighty, it's mito. But mitos are threads. All right? Because mitochondria, you always see them as these little oval-shaped things, right? But they don't have a wall or anything. They're just membranes, so they kind of stretch out and squish up and stretch out and squish up when they're inside of a cell. And so, you know, microscopists were looking at them, and some of them were kind of stretched out and all stringy looking, so they called them mitos, right? Mitos and chondros, threads of grains, right? Because they mostly look like little grains, right? Little thread-like grains. Then they carry out cellular respiration, which is how we get most of our what kind of energy? ATP in particular. Yeah. Chloroplasts, they do photosynthesis, which means they take light energy and use it to make food, make carbon compounds. Right? And there's also peroxisomes. Peroxisomes, they have enzymes called peroxidases. All right? Peroxidases break down hydrogen peroxide. All right? Why is hydrogen peroxide there?
Isn't it some byproduct? Right?
Yeah, it can form free radicals. It's a byproduct just of living in an oxygen atmosphere. Right?
But doesn't it also, like, because it destroys it, but it also has, protects the rest of the cell from it as well?
What does?
So the peroxisome, it
Yeah.
destroys it, but it also protects the rest of the cell from it.
Yeah, that's why it destroys it. Yeah, so our cells are full of water, and we live in an atmosphere that has oxygen. And when you combine the two, they actually form. All right, so just by living in an oxygen atmosphere, there's peroxides around. That's why obligate anaerobes are obligate anaerobes. They don't have the enzymes to break this down. And so if they're around oxygen, there's peroxides and free radicals everywhere and destroying their molecules. Right? I'll tell you guys later what free radicals do. It's bad. Very bad. Like decapitating your phospholipids. Think about that. All right. So the number one thing that makes a eukaryote, the nucleus. All right. The nucleus stores our chromatin, which will condense into chromosomes. Yeah. Stuff goes in and out of the nucleus because it has nuclear pores. It also has a double membrane. See, there's two phospholipid bilayers. There's a double membrane around it called the nuclear envelope. Right? It also has a very dark staining structure called the nucleolus. Do you guys know what the nucleolus is for? You're making ribosomes. Yeah. They're called ribosomes because they're made of. They have ribose because they're made of RNA. Yeah, they're actually RNA and protein, but the RNA for ribosomes is being synthesized here. And how many ribosomes do your cells need? How many proteins do your cells need? Lots. How many ribosomes do your cells need? A lot. So you're always making lots of ribosomes, so it's very dense. Right? It's probably the highest area of gene expression that you have in your cells. Yeah, so lots of ribosomes being made. And then once they're synthesized, they get out of the nucleus into the cytoplasm through the nuclear pores. Yeah. Ooh, black and white color highlight. All right. So then it actually connects to the nucleus. You guys see that? The outer membrane of the nuclear envelope connects to the rough ER, which connects to the. That's not the Golgi. That's the smooth ER. Yeah. So all these and the endomembrane system, as it's called, is all connected together. It's not that it all evolved from infolding of the plasma membrane. and then part of it eventually surrounded the chromosomes and gave them their own compartment. And then you have a eukaryote. Eukaryotic evolution. All right, but yeah, so the nuclear envelope and the ERs and even the Golgi, they're all related to each other, right, evolutionarily. So the smooth ER, that's where you make lipids. Ooh, got 1 point, just remembered. Where were we? There's something I didn't mention. Oh, here. So these guys, they don't just destroy peroxides, they also break down fatty acids. Okay. So when your cells are burning lots of fat, your peroxisomes are having fun. All right. So the smooth ER, it modifies things, and that usually makes them less toxic. But it also where you make your lipids, phospholipids, triglycerides, sterols. They're synthesized in the smooth ER. All right. Now realize that the smooth ER, right? It's a membrane structure. So when you make the lipids, they're actually in the membranes. Okay, the enzymes are in the lumen, but as the lipids are made, they basically diffuse into and embed themselves in the membrane. Right? So later on, when little vesicles pinch off, the vesicle itself has the new lipids in the membrane of the bubble. All right? Yeah. So. You can make lipoproteins, add lipids to proteins, you make glycoproteins, all right? So some of those modifications start to happen there, but a lot of the additions to your lipids and your proteins happen in the Golgi. Yeah. So here's an electron micrograph, see the rough ER, all right? So see how the rough ER is kind of flat? I mentioned that earlier, but it's kind of flat, and it's got the flattened lumen inside, and then you got the more tubular lumen inside the smooth ER, right here. Sliced into it, see the little openings. Ribosomes. Your book only has an illustration of 70S ribosomes, but you know, eukaryotes have 80S ribosomes. But these are protein factories, all right? Some can be membrane bound if the protein is going to go into the ER, and then the ER vesicles will carry that protein somewhere else. All right, somewhere besides the cytoplasm. If you need cytoplasmic proteins, you're going to use the free ribosomes that are just dissolved in the cytoplasm. All right. Interestingly, chloroplasts in plants and algae and mitochondria in all eukaryotes have 70S ribosomes. They have their own ribosomes.
I have a question.
Yeah.
Before you mentioned how antibiotics should be attacking 80 S, only 70 S, but if our mitochondria contain 70 S,
They don't get in our cells. Most antibiotics don't get into our cells.
oh, I see.
Yeah. They penetrate I don't know how, but they penetrate bacterial cells much better. But some can affect your mitochondria. There are some antibiotics that will make you a little bit sick because they affect your mitochondria. All right, the Golgi. All right. The Golgi have these big stacks. They're separate. You see how they're separated? They aren't interconnected like the ER is. But they're separate stacks, and there's a Golgi lumen. All right. And what happens is vesicles from the ER fuse, and that's what makes these big storage compartments of the Golgi. So each flattens. They're like little pita pockets, right? I said this last semester in my class. One of my students was like, "What's a pita pocket?" I'm like, I can't talk to you. Anyway, so it fused, and there's enzymes in there, and the lipids and the proteins that come from the ER vesicles can be modified. You can remove pieces, add functional groups, add carbohydrates and things like that. But there's a whole bunch of enzymes in there. And the chemical modifications they do, that, like I said, that becomes a destination tag, and those with the same destination tag, they pinch off in the same vesicle, and they go, you guys are going to the membrane, all right? You're going to, I don't know, this is somewhere else in the cell. But anyway, these big storage pita pockets are called cisterns, which literally just means like container. Yeah, so ER for the Golgi, so that's the post office of the cell. Oops. Any questions about the Golgi? It was discovered by a guy whose last name was Golgi, that's why it's called that. Italian scientist. Yes.
So if they pinch them, how does it regrow again, regenerate?
Oh, it's a continuum. I like to say that word. So there's vesicles always coming from the ER, and the vesicles fuse. So the vesicles fusing is what actually makes the cisterns. Does that make sense? So all these vesicles come, and like a vesicle hits another one, and it makes a little cistern. Another vesicle hits it, and it gets it. And basically, the vesicles coming from the ER form the cisterns, and they scoot forward. And so as a cistern, when it gets to a certain size, it kind of scoots forward. The cistern out in front is the oldest one, and it starts to break down to send its contents elsewhere. And so the front one gets degraded as new ones are forming in the back. Pretty cool, huh?
That's the mailman, not the post office.
The post office,
Post
yeah.
office.
Yeah. Yeah. So the mail's coming in, mail's going out. All right. And it doesn't look the same though. Hopefully your mail looks the same when you get it. So last couple things as far as the big organelles. So there's vacuoles. Like I said, you really only find these in this little piece of it here. Well, that's the nucleus. Okay. Yeah, you can see a little part of it, I guess. But the vacuoles, it's an alga. They don't need as big a vacuole because they aren't fully multicellular tissues. The vacuole gives support, right? Gives firmness to the cell, tonicity we call it.
Is there one vacuole or can there?
It's usually just one.
Okay.
Usually just one. Lysosomes, basically when endocytosis happens, endocytosis will bring something in, and a lysosome sitting there and it's got digestive enzymes in it. It's got proteases to break down proteins. It's got nucleases to break down nucleic acids. It's got glycosidases to break down polysaccharides. And it's got lipases to break down lipids. And so here's a food particle. Here's the lysosome. They fuse and it releases the enzymes into it, right? Because membranes fuse because they're fluid, right? And so then it digests everything. So we'll talk about that more later this semester, but that's essentially how it works. All right, so a couple important notes about the main guys. So the mitochondria, they have an interesting structure. They have an outer membrane, and then they also have an inner membrane. And the inner membrane is very highly folded. And why do we fold membranes? More surface area, okay? So the inner membrane has a lot of surface area because a lot of stuff happens in that membrane. There's a ton of enzymes and transporters in the inner membrane of the mitochondria. When it folds in, see how it folds in, kind of like a little cliff sticking off of a hillside or something like that? What do we call a little cliff sticking off a hillside? A crest, right? And so it's given the Latin word cristae, which means little crests, right? These little protrusions, all right? So those folds, and actually look how thin they actually are in a real mitochondria, right? So that gives lots of surface area. There's lots of enzymes, lots of pumps, lots of membrane transporters that all help process the energy that mitochondria take care of. We'll spend almost the whole next chapter on mitochondria, so you guys will learn all kinds of stuff about it. But what that does is you have two membranes. What that does, it gives you two compartments. So there's this space here. This is called the intermembrane space. It's very important. All right. There's also a space in the middle, and this space in the middle. So like there's a lumen here, right? There's a lumen in the middle, and the lumen in the middle is what we call the matrix. Even though the line is not going where it's supposed to. It's supposed to be the space in the middle. All right. See, this is the matrix right here. And there's all kinds of enzymes. Mainly the enzymes for the Krebs cycle are in here. That's where the Krebs cycle happens. You've probably heard of that before. You guys will learn more than you ever wanted to pretty soon about the Krebs cycle. But yeah, so that's the matrix. It's very important. Most people don't know that the matrix exists. It's where Keanu hangs out. Yeah, so lots of oxygen usage and lots of production of ATP, and as a byproduct, carbon dioxide. Yeah. So the inner membrane of the mitochondria has the oxidative enzymes that do respiration, and the cristae are what help make that membrane form. And then the chloroplast, called chloroplasts because they are green structures. But chloroplasts have a pretty interesting structure. They also have an outer membrane and an inner membrane. And the inner membrane folds into these little, even smaller pita pockets. And they also have spaces inside them, which we call lumens. Yeah, luminae is actually what you would call it in Latin. Luminae. Anyway, these are called thylakoids. All right, thylakoid comes from the Greek word that actually means little sacs, because they're little speed pocket sacs. All right, but the thylakoids are where photosynthesis happens. That's where it happens, people. The outer membrane and the outer part of the inner membrane, these are pretty much clear. The chlorophyll is in the thylakoid membranes. That's where the light energy gets absorbed, and then and processed and stored inside the lumen, and then the energy is transferred out into the space between. Does it have it labeled? Well, there's the thylakoids. They stack up and they form little grain-like things, so they're called grana, granum singular. But the space between is called the stroma. It's the liquid stuff that's strewn between them. But the thylakoids harvest the light energy, and the stroma has the enzymes. It has Rubisco for using that energy to make carbohydrates. All right, that's how photosynthesis works. Like I said, we'll learn more about it later on. All right, so it provides organic food for all living things on the planet. Poco importante. All right. And yeah, so the grana are the stacks of thylakoids. That's where the light capturing happens. And the stroma is the fluid matrix around them where the actual synthesis happens. So here's the photo. That's the synthesis. All right. So, last couple things for today. Mitochondria and chloroplasts. There's something interesting about them. There's a lot of interesting about them. They have two membranes. Why do you have two membranes? Where'd you get that other membrane? Guess what? They also have their own DNA. And as I told you, they also have their own ribosome. And what size are the ribosomes? 70S. And they have their own DNA. Guess what shape it is?
Circular.
It's circular. All right. Because they have their own DNA with their own genes and their own protein factories, they can actually make some of their own proteins. They're kind of independent. They also divide at a rate independent of how fast the cell is dividing. Right? Often faster. You often have dozens, if not hundreds, of mitochondria in your cells. All right. So, what's a possible explanation? Well, it's called the endosymbiosis theory, right? It's all one word. I just hyphenated it to separate the parts for you. Biosis, because things are living, and they are living together, together, all right? And they're living together where one of them is actually inside the other one, all right? So, last point. So it looks, all the evidence points to the fact that there was a large cell in ancient cellular history that engulfed a smaller cell. And that's how the cell got two membranes. And the smaller cell was highly energetic. It was either photosynthetic or a respiring cell. And the big cell's like, oh, internal battery, I kind of like this. So instead of digesting it, it just kind of kept it there. And the two eventually adapted to each other, but they started dividing at about the same rate over thousands and even millions of years, all right? And pretty soon, the cells kind of lost independence. They became dependent upon each other because this one lost some functions of cellular respiration, the other one was taken over, and this one lost some functions that it didn't need to absorb, didn't need membrane channels that much anymore, things like that, didn't need them, lost those genes because it didn't need them. That's what happens in evolution. If a section of DNA is useless, it will get randomly lost over time. All right? If you don't keep track of things, they get lost, right? You guys know this. But it formed the double membrane, and basically, if it was photosynthetic, this became the ancestor to chloroplasts, and if it was a high respiratory organism, it became the ancestor to mitochondria. All right, so that's the endosymbiosis theory. So basically, the big one provides protection and absorbs surface area to the little one, and the little one gives energy to the big one. All right, and we'll finish up looking at more details about the evidence for this. But this is one of the